of us are ready. Laura, do you want us to proceed?
Thank you very much, Juan Ramon. Good morning, all. I'm very pleased to introduce this webinar that deals with health and well-being. I'm very, as I said, I'm very pleased to organize this CU Talent event. Behind this organization, there is a great team that are participating in this project, in Research project, and in this initiative, the CU Talent initiative, we are trying through different webinars of different topics to first disseminate the research of junior researchers, and also to provide you a platform, a way to be in touch and to maybe promote and support collaboration, research collaboration on different topics in this CU Alliance, where we have 6 different universities with the incorporation of 3 more, and it's also having some impact in other universities beyond this alliance. So we really hope that this initiative can help junior researchers to proceed with and to go on. Okay, I think there is something, I can hear something at all your microphones off.
Laura, please reactivate your microphone.
Have you heard me before? Yes, it's okay? Okay.
Yes, now it's okay.
Okay. Well, today, I would like to introduce my colleague, Carmen Castro. She is full professor at the University of Cádiz at the Department of Biomedicine, Biotechnology, and Public Health. She belongs to the Institute for Research and Innovation of Medical Sciences, and she's performing research in the field of neurobiology. Some of her current research projects in which she's involved, and some of them she's a principal researcher, she's coordinating one of these projects, are dealing, for example, with searching for new drugs for the treatment of human glioblastoma. I don't know if I said it in the right way. And for example, a very interesting research, which is the relationship between diabetes and Alzheimer's diseases. Well, she's going to chair this session. And I would like to give the floor to Carmen Castro. I hope you enjoy this CU Talent event, and I invite you to participate as speakers and as participants in next CU Talent webinar. So the floor is yours, Carmen. Thank you very much.
Well, thank you, Laura, for your presentation, and welcome everyone to this CU Talent Talks on this so interesting topic of health. We're going to have 5 different speakers from different CU universities. And our first speaker today is Soizic Garaud. I hope I said your name correctly, not very good at French. And she's a PhD in life science and health from the University of Brest. She's an immunologist, I guess, and she is now doing her second postdoctoral position. And she will talk about immunotherapy and the secondary effects of immunotherapy. So just welcome to the meeting, and you can start, Soizic.
Okay, hello.
Excuse me, just one thing. We will talk for 15 minutes and the questions will be at the end. Okay, just, I'm sorry, Soizic, you can start now.
Okay, hello everyone. Can you see my screen?
Yes, we
Yes.
can.
Okay. So thank you for giving me the opportunity to present my project today. Started 1 year ago at the University of Brest on predictive biomarkers of immunotherapy side effects. So this project is supported by the Bienvenue program, co-founded Marie Curie. As you probably know, the past decade was a new age of cancer immunotherapy, as illustrated here by these different covers in prestigious journals such as Science, Nature, in 2014 and 2015. With this new idea that is not the disease, but the immune system that is targeted. Advances in understanding of cancer immunology have added immune escape as a hallmark of cancer, as you can see here in pink, and dramatically changed the way to treat cancer patients. The work of Professor Allison and Professor Anjou, who received the Nobel Prize in Physiology or Medicine in 2018, demonstrate how to manipulate the immune system by targeting immune checkpoint to reactivate the immune response. Indeed, cancer cells use immune checkpoints to escape the immune surveillance. So ipilimumab, an anti-CTLA-4, or nivolumab, an anti-PD-1, can reactivate T cells to kill cancer cells. So clinical trials have demonstrated the benefit of this drug, first in advanced melanoma, but also in non-small cell lung cancer. So this Kaplan-Meier curve shows you in blue patients treated by ICI, immune checkpoint inhibitors, compared to a control group. So in red, it's patient treated by placebo, and in green, it's patient treated by chemotherapy. And as you can see, we can these patients treated by immune checkpoint inhibitors have a longer survival compared to the control group. So now immune checkpoint inhibitors have been approved for several cancer indications by the FDA, but also by the European agencies, of course for melanoma and lung cancer, but also for hepatocellular carcinoma, gastric carcinoma, renal cell carcinoma, cervical and urothelial cancer, and more recently for triple-negative breast cancer. However, this drug can provoke inflammatory side effects called immune-related adverse events. So these ARES are characterized by a wide spectrum of clinical manifestation ranging from mild to severe. So as you can see with this cartoon, the most frequent one are anemia, skin toxicities, colitis, thyroid dysfunction, and less frequently, but more severe, myocarditis, pneumonitis, and neurological disorders. So IRAS usually occurs after a few weeks or months after treatment initiation. So usually skin toxicity and thyroid dysfunction occurs very soon after the initiation, but they can also occur at any time and even after treatment cessation. So it's really, really challenging for clinicians and especially for oncologists to diagnose this IRAS. Regarding the pathogenesis of these toxicities, several mechanisms have been proposed based on clinical observation and very small studies, such as an activation of self-reactive T cells, an induction of autoantibodies, the secretion of pro-inflammatory cytokine and chemokines, such as IL-17, Some reports also suggested an association with genetic factors and the microbiota. It's important also to know that some cells, such as pituitary cells, express immune checkpoint constitutively. And when we treat patients with anti-CTLA-4, so ipilimumab, you can target this cell. And in this case, we talk about an off-target effects. However, the physiopathology of these toxicities remain largely unknown. That's why the aim of my project is first to decipher the immune response of patients who experience RS using several discovery cohorts in order to identify immune biomarkers that can predict the onset of RS. And for this second aim, I will use a validation cohort. So first, for the discovery phase, I'm currently collaborating with clinicians at UZ Hospital in Brussels to collect samples. So here we use samples from the ECIT clinical trial. So it's a prospective clinical trial enrolling cancer patients treated by immune checkpoint inhibitor in standard of care. And the goal is to collect blood samples at baseline, but also at the onset of RS. So this cohort is composed of melanoma patients, mainly, but also non-small cell lung cancer, squamous cell carcinoma, renal cell carcinoma, and bladder cancer. And most of the patients were treated by anti-PD-1 alone, as you can see here in purple, or in combination with anti-CTLA-4. And the most common toxicities are skin toxicities, thyroid dysfunction, and liver toxicities. And the goal of this project is to characterize the immune response. So here we use circulating cells and perform flow cytometry analysis. And we also quantify soluble markers such as cytokine and immunoglobulins. So here, this heat map shows you some changes at the onset of RS compared to baseline, such as T cell changes. So in T helper or cytotoxic T cell subsets, or in B cell subsets. At the top, you can see some clinical information, such as the type of cancer, the type of treatment, the organ affected by the RS. And as you can see, there is no clear association with these clinical parameters. As we have an interest in B cells, we also try to characterize the maturation stage of B cells. So I will not enter in detail. I know you are maybe not familiar with immunology, but just to tell you that we observe an increase of a terminally differentiated B cell at the onset of RS, including activated memory B cells, but also antibody secreting cells. Then we also analyze cytokines and autoantibodies in the plasma of patients, and we observe an increase of cytokines, including interferon gamma, IL-7, IL-10, IL-8, and MIP-1 beta. Interestingly, we observe that 29% of patients were positive for at least one autoantibody at baseline and also at the onset of RS. So the presence of this autoantibody at baseline suggests the presence of pre-existing autoantibodies. Then, to go deeper in the characterization of the immune response, I'm currently establishing another clinical trial here at the University of Brest. It's a retrospective clinical trial enrolling cancer patients treated by immune checkpoint inhibitor standard of care, and with available tissues of affected organs. And here the goal is to characterize the immune response within affected organ using imaging mass cytometry. So imaging mass cytometry enables us to investigate 40 parameters on one paraffin section. And this approach or technology is available here in Brest at the Hyperion platform hosted in our lab. So to date, I've collected skin biopsies from patients who experience ICI-induced skin toxicity. Here it was lichen planus. So lichen planus, it's an inflammatory skin disease characterized by the disruption of the basement membrane, and the infiltration of immune cell, as you can see here with this purple dot. At the same time, we use a control group of patients with a wild-type lichen planus, so without any treatment. And using imaging mass cytometry, we were able to analyze the immune infiltration. So here in cyan, you can see epithelial cells in both groups of patients. So on the left, it's ICI-induced lichen planus, and on the right is the control group. We also observe the infiltration of T helper cells in green and the presence of cytotoxic T cells in yellow in both groups again. But what is interesting is the infiltration of B cells here in red in the ICI-induced lichen planus. And B cells were not present in the control group. So to go deeper in the characterization of this immune microenvironment, I've developed a pipeline using deep learning algorithms in the VisioFile software. First, to detect the tissue, then to segment it. So here you can see the epidermis and here the dermis. Next, to segment cells and then to phenotype these cells. And finally, I've also created another app to visualize B cell hotspots. So using this approach, we were able to identify 7 clusters of cells using this TSNE visualization. So including, of course, epithelial cells, T helper, T cells, cytotoxic T cells, macrophages, and B cells here in pink. We also perform neighborhood analysis and clearly observe a close proximity between immune cells, especially between macrophages and T cells. As mentioned previously, we have an interest on B cells, and here we quantify the density of B cells in both groups of patients. And again, you can see here an increase of B cells in ICI-induced lichen planus. And on the right part, you can see the distribution of these B cells. So here's the distance between B cells and other cells. And you can see here, this distance is very, very short between B cells and T cells and also macrophages. This is not the case with epithelial cells. So demonstrating really a close proximity of these cells and maybe an interaction between these immune cells. So as mentioned at the beginning, the ultimate goal is to identify predictive biomarker. And to do that, I will use a validation cohort named TADIC. TADIC, it's a prospective clinical trial here at the University of Brest. So this trial will start at the beginning of this year, and we will enroll cancer patients treated by immune checkpoint inhibitors alone, or in combination with conventional treatments such as chemotherapy, radiotherapy, and targeted therapy in standard of care. And the goal will be to enroll 200 patients in 2 years and to collect blood at different time points. First at the baseline, of course, so before treatment initiation, but also during the treatment. So after 3 months, after 1 month, after 3 months, and after 1 year of treatment. And of course, if this patient experience toxicity, we will take again blood samples. And then we will follow this patient during 2 years in order to collect clinical data and to see the evolution, the outcome of this patient. So finally, I would like to thank all members of the LBI team, my collaborators, so clinicians, and my collaborators in Brussels, the Hyperion platform, my fundings, and finally all patients and their family for their contribution in this clinical trial. So thank you for your attention.
I think, Carmen, that we cannot hear you.
Sorry. Thank you very much, Soizic. That was a great talk. We will wait till the end to ask you some questions about it. And now our next speaker will be Anthony Herzig from University of Brest. I will talk about genetic panels. He is a postdoctoral researcher I'm sorry, yeah, at the University of Brest. He did his PhD in Paris, at the University of Paris Diderot and University of Sorbonne. And he will talk about how our genetic background influences the development of different types of diseases. So, Anthony, you can start.
Hello, everyone. Can you hear me?
Yeah.
Okay. Well, thank you very much for the introduction, and thank you very much for the opportunity to discuss my work. So just briefly about me, my background is in fact in mathematics and statistics, but then, yes, I began my PhD in public health in Paris, and since 2019, I've been a postdoc in Brest in France. And so this talk, the domain is genetic epidemiology. So just a brief introduction of that. So we each have 23 pairs of chromosomes, each one consisting of a very long DNA string assembled from 4 different proteins labeled A, C, T, and G. And of course, we all have roughly the same thing, but there can be a few points on the genome where we can have a variation. People can have different base pairs, so we'll call these genetic variants. And we each have about 4 million of these genetic variants, and we each have about 70 new ones, new in the sense that we did not inherit them from our parents. And well, the most part of these genetic variants are very likely to be completely neutral. For public health, for example, we know on chromosome 17 there is the gene BRCA1, which we know to play an important role in the risk of developing breast cancer. So how do we go about finding which genetic variants are important for public health? Well, so we can either study families, so look at family trees and look at which genetic variants segregate with the affected individuals with a certain disease within a family, or we can have more things like case control designs, when we look at differences in frequencies of certain genetic variants between groups of case individuals and control individuals. So that's pretty simple if we can agree, but there are some quite significant challenges. We'll go through a few of some of them here. We have problems with data quality, so we often have lots of missing data, erroneous data, and in general, genetic data is quite expensive to generate as well. We can have batch effects where data coming from different sources are often hard to analyze jointly. As mentioned, we in general have very little idea of what most variations in the genome do, if anything at all, if they have any biological impact. And what I will focus most on is this idea of population stratification. So this is the idea that certain genetic variants might have quite different frequencies in different parts of the world, basically, due to historical population migrations and the ancient population migrations in the human population. So here we have an example of a variant which has very different frequencies in different parts of the world. It's actually a variant which plays a role in determining how well individuals can digest lactose, for example. And so in order to take this into account, it's very important to have panels of kind of reference individuals, so large databases of individuals from different populations, so we can know what genetic variants are out there and what their frequencies are. So most well known would be something like the 1000 Genomes Project. We have individuals from across the world who have been sequenced, so we can catalog different genetic variants, how frequently they occur in different populations, and then have an idea of which ones might play a role in public health. Estimating the frequencies of variants is very important for all sorts of statistical modeling, particularly as we have this kind of idea often in genetic epidemiology that if a genetic variant does have a biological impact, we can often expect a kind of inverse relationship between its frequency and its impact. The idea being that variants which are observed to be very frequent in the population are unlikely to have a high impact, because if they did have a high impact, their frequency would often be attenuated due to selective forces. Okay, so I'm going to go a bit further into this idea of population stratification. It's this idea of when you have kind of systematic differences in allele frequencies between subpopulations. And we can assess this basically by combining data across many thousands of genetic variants in order to describe the structure of a population. So, for example, the principal component analysis, where we find axis of most variation using statistical modeling. So here you can see very vaguely some points. They'll become a bit clearer in a minute. So each point here represents a person, this is a principal component analysis of their genomes. And you can only vaguely see what's going on here, but I'll add these crosses which describe the kind of distribution of the cloud points of individuals coming from different European countries. You can see that essentially we find, again, the map of Europe in this kind of analysis. Okay, now, just very important to note, this is not to say that there's any really significant difference between the genetics of someone coming from Finland or someone coming from Sardinia. It was important to look at this well-known result that, in fact, between a pair of individuals, their genetic differences, if they're coming from the same population, can often be higher than if they were coming from different populations. So it's not to say that everyone coming from Finland are very similar to each other, or very different people from different countries. But the idea is by studying many individuals together and lots of different genetic variants, we can nevertheless infer these very subtle genetic trends coming from ancient population migration. You can see there's quite a gap in the middle here.
So this can be filled in by sequencing individuals from French populations. This is what we're working on at the moment in Brest. So you can see a project I've been working on, which is the France GenRev project, where we sequenced 850 individuals using blood samples. And you can see they fit onto the map quite nicely here, with the individuals from the west of France close to the individuals from the United Kingdom, the individuals from the east and north of France are close to the individuals from Belgium and Germany, and from the south, we're closer to the Spanish or Basque populations. Now, you may be wondering how it is that I can place my individuals on the map of France, and we do that basically using a particular recruitment process where for each individual, we asked them where their 4 grandparents were born. And in fact, we selected individuals who had their 4 grandparents all born close to one another. So then we could basically geolocalize each individual on the map. And this kind of study design allows for some really a sort of interesting potential analysis of the structure of the population. We can go right down to fine scale levels structure. For example, here looking at the west of France, we did a clustering analysis, and you can see we separate individuals who have very slightly more similar genomes to each other, which correlates very well with the geography. We even have these 2 groups here with a boundary that follows quite closely the Loire River, which is the largest river in France. Okay, so why are we doing this? Well, as I said before, this is the idea of simply finding individuals from the general population to build genetic reference panels, which we then use in many different ways for genetic epidemiological studies. So before I mentioned that we each have about 4 million genetic variants. So here I have my individuals, and I've classed them based on how many genetic variants they have, going from the least to the most. And you can see basically, well, no pattern at all. However, if I then filter out any variants that have previously been seen in other studies, so this is a huge database of genetic variation across many different populations. If I look at only the variants which have never been seen before, so these ones that may be more particular to French populations, you can see a clear pattern. And this is happening because the existing databases at the moment are very enriched in genomes from the north of Europe and from the United Kingdom. So more likely the variants which are in the west of France, we have already seen them in one of those populations before. Hence why we need to have more individuals from, for example, from the south of France to provide a reference panel for these populations. So at the moment, we're making available catalogs of the genetic variants that we found, with describing their frequencies And this becomes a very important tool for filtering out variants when studying in more sort of clinical settings, when trying to decipher which variants are important for different diseases. And of course, this data set can also serve as a control cohort. So if other researchers have sequenced case individuals, they can collaborate with us to compare their case individuals to our controlled group, to allow for the study of the association between genetic variants and different multifactorial diseases. Another very important use of genetic reference panels is for imputing missing genotypes. So we often have this set up. So for example, the individuals of that project I was just discussing, we have high quality whole genome sequencing data for these individuals. So we have a good measure of their whole genome. But very often in other studies, what you might do is you might only record data on a few sparse genetic variants at certain locations on the genome, because this is a lot easier to do and it's a lot cheaper to do as well. But due to the fact that we all have very similar genomes in general, by comparing the similarity between across these variants which are in common between these two groups, we can actually very accurately infer all of these missing genotypes and add a lot of information to different data sets. So this is something I work a lot on, methods for doing this kind of missing genotype imputation. And again, we show why it's very important to have basically reference individuals from lots of different regions, because, for example, if we impute the individuals I described before, we get exactly the same kind of pattern where individuals from the West and the North can be imputed with a lot higher accuracy. That's because existing genetic reference panels are, as I said, very enriched in individuals from the North and the United Kingdom. But we impute much less well the individuals from the south of France. Hence why we are currently in Brest, led by the team in Brest. We are undertaking a new national project. We'll again be recruiting individuals using this design of asking people where their grandparents were born. These are in fact individuals from existing epidemiological cohort constants. And among about over 100,000 people, I selected 15,000 individuals, looking for people who had 4 grandparents born near to one another. This time we're kind of covering the whole of the geographical region of France. And these individuals were all sent saliva collection kits through the post, and the DNA extracted from those saliva samples is currently being sequenced in Paris. So we have some early results from this data set. So for about 9,000 individuals, here are the longitude and latitude of these individuals from the grandparent birthplace data. I've colored the individuals based on old administrative regions of France, and the small boxes with numbers refer to different administrative departments of France. And then taking genetic data from these individuals, we can perform analyses looking at sort of the fine scale structure of this population. So this is in fact similar to the TSNE visualization that Soizic just presented a bit earlier, but this time with human genomes. And we can see that we find essentially we can find the map of France, but just from the genetic data of these individuals, showing why the fact that we will have this group as a reference panel will allow us to really catalog all the different genetic variants going on in some French populations, and this will also again provide both an imputation reference panel and a control panel for performing case control studies. And the data will soon be included in a wider pan-European 1 million genomes initiative, so combined with other populations. And indeed, it can also further be combined with populations from other parts of the world to better our sort of catalogs, our databases of human genetic variation. Personally, I'm working on some method developments for working out if our control individuals have been chosen in this way based on geographical criteria. How can we correctly compare them to case individuals who of course won't necessarily have their 4 grandparents all born near to each other, and they might have parents from different regions or even different countries. And we basically developed a method where for each case individual, we kind of model their parental genomes in order to create a pseudo control for each individual in order to perform a statistical test looking for association of different genetic variants with the case status, but which takes into account this complicated population structure or population stratification. And then just a bit more about our lab in Brest. So I mentioned earlier that the individuals of PopGen, we sent them saliva collection kits. So we've been doing a lot of work on assessing how good the quality of genetic data is when we use saliva instead of blood, because blood is typically blood sample would be typically the way to go. And what we found is a really nice result is if we use saliva, we can get at the same time good quality human genome data, but we can also characterize individual salivary microbiomes. So for those individuals from PopGen, we will have eventually both a good measure of their genetic variants, but also we can characterize different bacterial populations potentially. My group is quite focused on sort of statistical method development, so developing ARC packages for statistical methods in genetic epidemiology. For genetic data management, so genetic data is often very large and difficult to work with, as well as being very sensitive data. So working on methods regarding genetic data protection and safe sharing of genetic data. So just to conclude, building genetic reference panels is very important for understanding and cataloging different human genetic variation. They can be used for imputing missing genotypes, which helps to really add a lot of value to a lot of different data sets. And for describing population stratification, all of this coming together to inform and improve statistical methodology for genetic epidemiological studies, with the kind of wider goal of improving public health by eventually being able to provide potentially better screening for patients. By finding which genetic variants affect which diseases, we can hope to provide new molecular therapies, and just to, of course, very important to improve public awareness of the role of our genomes. So yes, we're in Brest. There could be some, or there will be some postdoc positions coming up quite soon. So if you're interested in statistics, genetics, bioinformatics, microbiomes, don't hesitate to get in contact. And thank you very much. Happy to answer your questions a bit later.
Well, thank you. Thanks so much. If I were a statistician or a mathematician, I would definitely go to the laboratory to work. Next speaker will be Marcin Michalak. He has a background in law, and he is an assistant professor at Gdansk right now, and he's collaborating with Yale University too, and who will talk about health and law, and a subject that has been really important over the past 2 years, and how we need common law for the prevention of infectious disease. So, well, you can start now, Marcin.
Yeah, thank you very much, Carmen. I will try to share with you my presentation first. Okay, we'll just. Can you see this?
Yes, we can.
Okay, just a second, because it disappeared from my screen, but okay. So hello again. First of all, as the previous speakers, I would like to thank the organizers for giving me the opportunity to speak to you today and share my research project with you. And I would like to start by saying that the issue of coping with infectious diseases is eminently interdisciplinary, as we can experience and see very clearly today during this seminar. And as I can imagine, my presentation would be of completely different type than the two of which we have a great chance to see, but I hope you will like it. So the role of the physicians, the role of epidemiologists, virologists, biologists, and statisticians, etc., cannot be overestimated in the event of a pandemic. However, the COVID experience has shown how great is the role of also the role of the good legislation of law in effectively combating such health threats. So proper regulations of the use of protective measures, maintaining social distance, the obligation to vaccinate, or the role of so-called vaccination certificates cannot be, as I think, overestimated in the fight against the pandemics. So in addition, as the viruses, there are no national borders. For the viruses, there are no national borders, of course. The question of creating effective legal tools in dealing with the pandemic has become the subject of attention, not only at the level of internal legislation of individual states, which is obvious, but also at the level of international law. And from this perspective, the European Union law occupies, as I believe, special position for at least several reasons. So first of all, the existence of the common European markets, the free movement of people and goods, including animals and food, are directly related to the dynamics of the spread of infectious or communicable diseases on the territory of the EU. So, I'm sorry. So that's the first thing. The other is that giving the place that the European Union occupies in the international arena with its economic and scientific potential and legal culture, the community is somehow predestined to set good standards in a global system of prevention and control of infectious diseases. And last, and as I think not least, the specificity of the European law as a supranational law gives the European Union unique opportunities, but also poses a special challenges in the area of legislation for prevention and control of infectious diseases. So the question then arises, what is this supranational law? What is its specificity, and what is its potential in the context of communicable diseases law? So the supranational law is a form of international law, and it is based on limitation of the rights of sovereign nations between one and another. And the EU is probably the best existing international organization which applies this concept in practice. And the concept was characterized in a very plain way, as I think, by two very important rulings of the Court of Justice of the European Union from the 60s, namely the famous ruling in Van Henten Laws and Costa Enel. And what the court indicated then was that by contrast with the ordinary international treaties, the EU has created its own legal system, which became an integral part of the legal systems of the member states, and which their courts are bound to apply. And this separate legal system takes precedent over the norms of national law in the matters it regulates. In practice, it means that the EU supranational law is actually much more effective in practice than traditional international public law. It is so because all the national courts are bound to apply it on everyday basis, and all the individuals in the EU can derive its rights from the European norms. And just to give you the examples how it works in practice. For example, whenever you buy a product online, we can send it back within a minimum 14 days to the seller without giving any reasons. So we can simply return the product, and all Europeans in the EU have exactly the same right, which stems from the Article 9 of the Directive on Consumer Rights. So any of us in a possible legal dispute with the seller can invoke this European standard, and the national court in Spain, Poland, Romania, Netherlands will have to apply it for the consumer's protections. So the same mechanism applies in the area of healthcare. For example, in November 2022, very recently, the court said that the Netherlands does not have the right under the domestic law to expel a Russian citizen who is ill and is being treated in Netherlands with medical marijuana, because in the absence of appropriate medical treatment in Russia, the patient might be exposed to a real risk of rapid, significant, and permanent increase in the pain linked to that illness, which means that it is the European law, and specifically Article 1 and 19 of the EU Charter of Fundamental Rights, that stands in a way of expelling such person from the EU. So since we know that EU supranational law has a special power as international law, it is worth asking whether it provides any common solution when it comes to the prevention and control of infectious diseases. Here, the verifier was, of course, the COVID pandemic. So when the COVID began in Europe, first with great intensity in northern Italy, the European Union was not ready for the scale of the challenge. And as the EU Commissioner Johansson very aptly put it, in the crisis, EU member states acted like humans do. The first instinct being was to fend for themselves until they realized the value of cooperation and the potential of supranational law in this regard. So I believe that we can derive similar obvious benefits from good cooperation based on fine law in the European Union in the area of infectious diseases, as we do in, for example, in the area of consumer rights or patients' rights. And the coronavirus epidemic showed that there is much room for improvement at the EU level in this layer. So if we have been able to create a highly effective legal framework in this regard, we should be more cooperative and more coordinated in the area of communicable diseases prevention and control. So first of all, we have so far created some structures and institutions that make this possible. There is a European agency which named as the European Center for Prevention and Control of Infectious Disease, with the headquarters in Stockholm. But when this agency was established in 2004, it was just an advisory body with a little real competences when it comes to taking action in the area of effective response to pandemic threats. But when infectious diseases have become such a serious threat in the second half, in the next decades of the 21st century, the ECDC role in the health system preparedness and surveillance should be expanded. So basically, the aim of my research as a lawyer is how the European Union law on infectious or communicable diseases can and should evolve so that it can be more effective and better serve for the Europeans. So first of all, legislation may be needed to improve compliance with data reporting. So the voluntary nature of current surveillance initiatives leaves the ECDC without a real means to force member states which fail to meet reporting standards to do it better. So had we had timely and reliable data from Italy on infectious, perhaps the tragic effects of the virus spread could have been contained in time. So the idea, therefore, is to establish an effective and a uniform system for reporting on the current state of epidemic risk in member states, and for ECDC to supervise the correct operation of the system. A major success of the European Union level has been the joint purchase of vaccines. So the vast majority of us here are the beneficiaries of the fact that it was the European Union and not the individual member states that decided to negotiate and purchase vaccines from the big pharmaceuticals companies. But still, much remains to be done in this area as well. As you know, as all of you know, at the different stage of epidemic, for example, in Polish planes or airplanes, it was not necessary to wear masks. In German and Danish, it was. In Portuguese and Greek, in some circumstances, it was obligatory to wear masks. So we have an EU vaccination certificate, but the conditions of entering public places after its presentation dependent on the individual decisions of each country. So I think that as Europeans, we can afford to be consistent in this area, and this can be guaranteed by the European infectious disease law, common European infectious disease law. So we should develop European system of production and distribution of medicines, vaccines, and other medical countermeasures, such as gloves and masks, that were often lacking during the first phase of response to the COVID pandemic. And important developments that are also the recent legal acts enacted in the EU, especially the regulation from December on serious cross-border health threats and the regulation amending the regulation establishing European Center for Disease Prevention and Control. But many questions remain open regarding legal solution at the EU level to effectively combat the challenges posed by infectious diseases in the 21st century. However, I believe that so-called after COVID era represents a once-in-a-generation opportunity to develop a comprehensive strategy to infectious diseases across Europe, and exploring the potential and the legislative possibilities in this regard. I would like to contribute with my research to some point to this bigger project. So European health policy should be forward-looking rather than reactive to health emergency and pandemics. And the existence of the European Union law has so far shown that united we can be very effective, and that unification in many areas made possible by good law is done to the great benefits of millions of Europeans. So we now have a great opportunity to do so in the area of European infectious disease law as well. And I would like to add a little piece to this big project. Thank you very much.
Thank you very much, Marcin. I'm looking forward to the end to ask you some questions. And now our next speaker will be Brendon P. From University of Malta. His work previously at University of Amsterdam, and where he did his PhD in experimental medicine. So now you can start, Brendon.
Yes, good morning, or rather good afternoon now, and thank you for the introduction. Today I'll be talking to you about the past work and also current work that I've done and I'm doing with respect to understanding sepsis pathophysiology, in particular epigenetic properties of immune cells and how these properties may harbor important implications for the future of sepsis treatment. So what is sepsis? Sepsis is currently defined as the abnormal reaction to an infection that leads to lethal organ dysfunction. It's been recently, the epidemiology of sepsis has been recently revised, and it's estimated that worldwide, at least in 2017 alone, so this is before the COVID pandemic, around 48.9 million cases were reported and 11 million deaths, which makes sepsis a leading cause of morbidity and mortality on a global scale. In fact, this data has motivated the EU to recognize sepsis as a global health problem. So sepsis can be caused by various types of pathogens, including influenza, Haemophilus, Staph aureus, and also SARS-CoV-2. In fact, critically ill patients due to SARS-CoV-2 infection are diagnosed. This is now being recognized as viral sepsis. In addition, E. coli, Enterobacter, and also other fungi can cause sepsis, and all major organs can be impacted with quite a variable inter-individual response in terms of the degree and the type of organ dysfunction that is consequent to the infection. The major organs can be affected, and predominantly the respiratory system, so pneumonia being one of the major determinants of sepsis, and abdominal infections followed by genitourinary infections can cause sepsis. It's also fair to mention that age is a predetermining risk factor for sepsis. In particular, individuals with underlying comorbidities such as diabetes, hypertension, and COPD. In addition, also neonates, infants, can be affected by sepsis. So sepsis is quite indiscriminate in terms of its impact and the susceptibility spectrum. Through advances in medicine, we're now seeing that a lot of patients admitted to the ICU survive sepsis, which is a good thing. However, what we're also observing is that sepsis survivors remain at high risk of long-term complications and also poor outcomes. So this is what we're now regarding as the post-sepsis syndrome, with fluid retention, immune dysfunction, so persistent inflammation, cognitive decline. And all this precipitates to a proportion of these patients, sepsis survivors, who are also eventually readmitted to the ICU. So what about the pathophysiology of sepsis? Well, it is currently, the current paradigm centers on simultaneous pro and anti-inflammatory reactions. So on the one hand, in this figure, you can see the top portion being the acute phase, or the what we're calling hyperinflammatory phase. And this hyperinflammatory phase can also, of course, relate to poor outcomes, for example, via increased chronic low-grade inflammation, organ injury, and muscle wasting. However, on the other end of this immune function spectrum is the anti-inflammatory response. These responses, if sustained, can precipitate to immune paralysis. And this leaves pretty much the door open to viral reactivations, nosocomial infections, and failed tissue recovery. So in some experiments we've performed in the past, we've seen that stimulating a PBMC, so these are peripheral blood mononuclear cells, obtained from septic patients on intensive care unit admission, by stimulating them with E. coli-derived lipopolysaccharide, or LPS, the PBMCs responded with a reduced cytokine production when related to healthy controls. And the same patients, upon recovery, we saw that these PBMCs, the response of these PBMCs was recovered, at least in part. Important cytokines being IL-1 beta, IL-6, and also a TNF. So what this told us was that the cytokine productivity of PBMCs from septic patients is hampered, and this is attuned to a phenomenon that is called immune tolerance. So tolerance is regarded as one of adaptive programs induced in innate immune cells. And the four main adaptive mechanisms being differentiation, priming, trained immunity, which I will also touch upon later, and tolerance. So if you look at the graph here for trained immunity, see that the first insult, let's think these are monocytes. Monocytes will respond by producing cytokines. After a second insult, these monocytes tend to respond in a more enhanced manner. And this phenomenon or mechanism confers long-term memory to innate immune cells. In contrast with tolerance, we see that the first insult does, as predicted, induce a cytokine response, whereas the second insult is associated with a dampened response, as I've shown earlier. So these are the four main adaptive programs, in particular trained immunity and tolerance, which confer memory to innate immune cells. With regards to adaptive changes, these can be induced, and they're predominantly induced by non-heritable factors, and this is the environment. So the environment shapes our immune system. Throughout our lives, we are constantly exposed to either pathogens, which most of us luckily just breeze through it without feeling any signs of infection. We're also exposed to medications, vaccinations, also the food we eat, and of course our lifestyle. So all these inputs are relayed to our genome via what is known as the epigenetic mechanisms. And epigenetics is a study of how our cells control the activity that is in response to external cues. Importantly, these activities do not alter the DNA sequence that we inherit from our parents. So in essence, epigenetics is a mechanism, a field that connects our genes to the environment. And in the same way, it shapes our immune system by, for example, altering to the right. This is an example of a monocyte. And monocytes are quite plastic cells. They are very well armed to produce cytokines. They can also partake in antigen presentation, phagocytosis, and also in promoting immune tolerance. Depending on the external cues, these cells can shift into these various functional states. So the current idea is centered on the model that was proposed by Conrad Waddington in 1942, and that is cells that were epigenetic topologies determine how cells gain identity and also a functional state. In this example, I'm showing you, for example, hematopoietic stem cells that will either take on a macrophage identity or a T cell identity based on how they roll down this epigenetic landscape. And beyond this identity, a functional state. For example, a macrophage can either become a pro-inflammatory and antimicrobial, but could also be more attuned to tissue repair and anti-inflammatory responses. So there are various chemical and enzymatic processes that are involved in reprogramming cell epigenetic landscapes and in consequence their functional states. And mainly we've got epigenetic readers, writers, erasers. So these are enzymes that recognize chromatin and the various modifications on chromatin, for example, in this case, methylation. They can also write these methylation marks or erase them. Others recognize, for example, acetylated regions. And in doing so, these enzymes can shift our chromatin into two main conformations. That is a closed chromatin or an open chromatin. Open chromatin is associated with higher gene expression, whereas the closed chromatin, where our DNA is now more compact, it does not allow for entry of, for example, RNA polymerase to express these genes. In addition to enzymes, there are also RNA molecules that have been associated with the mechanisms in epigenetics. And these are mainly subdivided into two categories arbitrarily. And this is based on their length. So we've got long non-coding RNA. So any RNA molecule that is not translated into protein, which is longer than 200 nucleotides. Whereas on the other side, we have the small non-coding RNA molecules. Again, these are non-protein coding molecules, and they are smaller than 200 nucleotides. One of the most potent of these molecules being microRNA, which can fine-tune gene expression. So all these different factors influence how, firstly, our chromatin is modified, enabling the expression of certain genes and consequently functional states of a cell, and likewise including these new long non-coding RNA molecules. So one of the first reports of chromatin modifications in relation to, in this case, tolerance, was in 2007 by Ruslan Medsitov's group. What the group showed was that inflammatory cytokines, for example, IL-6, are certainly tolerized, and this tolerance phenotype was associated with shifts in chromatin landscapes in response to toll-like receptor ligands, in this case LPS. However, to the right, you'd appreciate that some of these genes are not tolerized. So these are actually enhanced, almost enhanced. And these are the non-tolerizable genes. And most of these pertain to antimicrobial or antibacterial mechanisms. So this is the first report where epigenetics can influence the outcome or the functional state of a cell, hence reprogramming these cells. More recently, a study, or various studies rather, on trained immunity, and trained immunity being a mechanism whereby the activation of the immune system, of the innate immune system, promotes an enhanced response to other challenges, to secondary challenges. And this is spearheaded by Mihai Netea in Nijmegen. And what Mihai's group have shown was that the properties underlying the trained immunity phenomenon mechanisms are tightly interconnected with the epigenetic landscape of these cells, where in a resting state, so this is a trained unchallenged cell, chromatin is mildly condensed, and there is little gene expression, in particular of cytokines. However, after restimulation, that is trained and challenged, these cells now exhibited a more open chromatin conformation, and thereby a more enhanced cytokine production profile. I've also shown, together with PhD students 2 years ago, that monocytes obtained from community-acquired pneumonia patients exhibited a tolerant functional state. So in this study, what we've done was We've included community-acquired pneumonia patients during the acute phase, so this was on hospitalization, and we followed them up for 1 month. We also included age and sex-matched controls. After purifying monocytes from these patients and control subjects, we then stimulated these cells in vitro, and stimulation showed that, stimulation with LPS, showed that while indeed the healthy, the gray bars here, subjects exhibited a high response in terms of their cytokine productivity, during the acute phase, pneumonia patients showed a dampened, a significantly reduced response, in particular for TNF, IL-1 beta, and IL-10. What was interesting is, besides the recovery monocytes, so these are same patients, only 1 month after hospitalization, that the monocytes were recovered in terms of their cytokine productivity, but not entirely. In this case, IL-6 was not resolved. We went on to try and understand the epigenetic connotations to this process, the immune tolerance. And what we found was that a signature of immune tolerance in human monocytes was derived by a multiomics integrative approach. Here what you see is a Manhattan plot where each chromosome was assessed for DNA methylation marks, each dot being a specific, in this case, DNase hypersensitive site. Comparing our control subjects to community-acquired pneumonia patients on hospitalization, so during the acute phase, there were some changes that also were significant to the genome-wide scale, namely a chromosome 22 DNase hypersensitive site. Using a multiomics platform, we then combined RNA expression profiles or RNA sequencing profiles from the same monocytes, DNA methylation profiles, and their cytokine production. We found that there were quite some significant correlations in between the three blocks of data, with, in particular, the chromosome 22 site harboring a transcriptional enhancer element. And this was distal to many of the genes that we found also differentially expressed in CAP acute patient samples to control subjects. And in essence, what we observed was that, in particular, for cholesterol biosynthesis genes, there was a correlation between methylation at this particular chromosome 22 site and the expression of disease, positive correlation, which was associated to an inverse. It was inversely correlated to the IL-1 beta response in this case.
We're going to have to finish.
Yes, the same inflammatory cytokines have been shown in organ dysfunction and monocytes of sepsis patients in another study by Lorenta Sorolla. We also showed that different non-coding RNA molecules are also differentially expressed in septic patients, in particular long non-coding RNA and microRNA. And this is also the platform by which we are now studying these molecules in isolation, given certain in vitro parameters, for example, tolerance phenotypes. Lastly, is this related only to innate immune cells? No, it's not, because we also see reports from CD8 positive T cells where T cell exhaustion is associated with what has been reported as epigenetic scars in this case. So in conclusion, reversible epigenetic changes can indeed support variations in immune cell functional states during infection and in sepsis. Utilizing this knowledge and in particular dysregulation of epigenetic landscapes during infection, we can envisage that this can facilitate development of new and much needed, since there is no current specific therapy for sepsis besides supportive care. Lastly, to enable this deeper understanding of these specific factors, non-heritable factors that shape our individual immune system is critical to understand how and when immune-mediated pathologies arise, and also the risk for severe reactions such as sepsis. Thank you all for your attention.
Thank you very much, Brendon. And now we're going to listen to our last speaker, and she is Emilia Terzari. She's a lecturer at the Department of Nursing at the University of Malta. She's a PhD in public health, and she's going to talk about how preventive medicine can be used to control infection. So it's your turn now, Emilia, you can start.
Thank you very much. Can you hear me?
Yes.
Okay. Can you see my slides?
Yes, perfectly.
Yes. Okay. Thank you. So hello, good afternoon to everyone. And initially, I would like to thank the organizers and Professor Alan Jago from the University of Malta for organizing this very interesting and divergent, quite a divergent stream within healthcare. It is my pleasure to present to you some work I have been involved as focused person on infection prevention and control, work that has been done in collaboration with the World Health Organization on identifying what are the core components for effective infection prevention and control programs at the national level. So the WHO, as you can see here, this is the manual on the left-hand side that you are seeing are the guidelines that the WHO has issued on what are the core components on preventing healthcare-associated infections within healthcare facilities in countries worldwide. They have been established and launched based on systematic reviews that are referenced here, systematic reviews and also expert opinion. And the core components are identified as having an infection prevention and control program at the national level. Normally, they sit within the Ministry of Health or in various countries, depending on the agencies, the national or other sources, but usually in the Ministries of Health. Having a program, having national guidelines to prevent healthcare-associated infections and to combat antimicrobial resistance. Having another core component is to provide education and training on prevention of healthcare-associated infections. Having surveillance in place to monitor based on key indicators and key pathogens to monitor the prevalence and incidence of healthcare-associated infections, monitoring and auditing and providing feedback to healthcare professionals based on the infection prevention and control procedures that are identified as key indicators, and also having an enabling environment to support the above core components that I mentioned, which identifies the workload, the staffing level, and the bed occupancy that is necessary, and having in place the resources, importantly, the resources, materials, and equipment. And overarching this, what we see here as multimodal strategy, overarching these core components is the core component of having a multimodal strategy in place, which has been, which encompasses, as I said, in order to implement any of these infection prevention control procedures, we need to have multimodal strategies in place. These are activities, procedures that need to be in place. So it is not sufficient or it is not adequate to have the resources, but we need to also have this different elements that I mentioned, these core components, in place in parallel in order to achieve sustained improvement and ultimately to reduce infections. So the core components for infection prevention control, or IPC as I might refer as an acronym, are 8 for healthcare facilities, and so based on the facility level, and 6 core components at the national level that countries worldwide, both high income, lower income, have to have in place in order to have robust programs for their healthcare associated infections. And these are based on evidence-based recommendations and also, as I said, expert opinion. Now, other than these core components, the WHO suggests and has issued a list of minimum requirements for IPC programs that are listed here. And the aim of this study that I will introduce and speak about is to identify a situation, to perform a situation analysis that explores what and at what level are these minimum requirements that will then lead to fulfill all the core components, the full requirements for IPC. IPC standards that are based as well, the minimum requirements are based on these core components. They provide minimum protection and safety to patients, not only, but to healthcare workers in healthcare facilities, relatives, and also visitors. And it is the starting point to build strong and effective infection prevention in the hospitals. And this work has been led by previous work that identified that there is a low uptake or a low establishment of such programs in healthcare, in hospitals. And it targets implementation at both country level, but also at the healthcare facility level. And these should be in place in order to be able and progress to the full core components. And here we identify the core components. So having a, So having a functional IPC program in place, and this requests that there is at least one full-time trained and specialized infection prevention focal point in a country, and that there is also dedicated budget specifically for the infection prevention and control program. Evidence-based guidelines need to be in place, but not only based on a copy-paste from international guidelines, but these need to be adapted to the local context. Education and training is core, where there needs to be training provided to all healthcare professionals. And also this should be at pre-grad, at the healthcare professional level, and also at postgraduate for having specialized trained professionals in the field of infection prevention. Having national group, technical group that develop plans for surveillance to establish surveillance program, automated and surveillance monitoring programs. Multimodal improvement strategy should be implemented for any of the interventions, procedures. Catheter care needs to have a multimodal improvement strategy that looks at and implements these in parallel, and also having a set of key recommendations that are being systematically monitored, and training and feedback is provided on it. This follows this work has been based on behavioral science initiatives and implementation science, which encourages the use of a cycle and stepwise approach to achieve implementation, such as we identified the challenge we had with not having core components in place, preparing for action. Step 2, we identified that we needed a baseline assessment, since many countries did not have implementation of such core components. We targeted a baseline assessment. And also step 4 of this stepwise approach is to evaluate the impact that will drive further improvement. We based this work on on a validated tool. It was a questionnaire that was developed by the World Health Organization, known as Infection Prevention and Control Assessment Tool. That was the roadmap to guide the actions. It is based on 6 main sections at the national level, based on each of the core components, in total, totaling 112 validated indicators overall. And the tool has been pilot tested, and it is a self-assessment tool which requests healthcare professionals or those specialized or infection prevention units within healthcare facilities undertake, or at the national level, they undertake a self-assessment. In this case, since this was a work that was done at the national level, the focal points were ministers of health mainly that we interviewed and disseminated the questionnaires at the national level within the Ministry of Health, as I said, through a global survey that was conducted in 2021. As a main objective, we aimed to assess what was the current situation of infection prevention and control in countries. We aimed to provide a global snapshot of the implementation of these minimum requirements for IPC programs, and also we wanted to track the progress and compare with a previous national assessment that had been conducted. As I said, this was done through a validated cross-sectional validated tool questionnaire, through a cross-sectional online survey. Separate piece were the interviews that followed up with ministers, and it was also disseminated in various languages. It aimed to have a broad representation of the WHO member states. 194 were invited to participate, and the sample goal was to reach at least 25% of the member states from each of the 6 regions and from each of the World Bank income levels, starting from low income, upper middle income, high income countries. And as I said, it focused on national focal points within governmental institutions responsible for, as part of their own IPC on infection control. And we engaged regional country offices also in this piece of work. And there was a specific portal developed online to track progress, and where participants could submit their data. This part is based on a descriptive data analysis that was performed, that has been completed at this stage, but further analysis is yet to be conducted. So here we see the country representation and participation. We had a total of 106. In blue, we see the countries that participated, and in gray, those where we didn't have representation. 106 member states throughout, achieving 55% of all the countries that were invited. We managed to achieve more than 25% in most of the regions: Southeast Asia, Europe, Eastern Mediterranean, Americas, and Africa, 22% in Western Pacific countries. World, based on the income level, also there was quite good representation there. So some of the key indicators that were assessed, and our data showed that overall, from the total number of countries, 100%, those only 3.8% met 100% of the national infection control program minimum requirements. So only 4 countries achieved that. And countries that there was a higher percentage, 48% met 75% of the national IPC program requirements, and 77.5% met 50% of the minimum requirements. So none of the countries that met all the minimum requirements, none of those 4 countries was a low or lower middle income countries. So you can identify the gaps that are still existing, since these countries were of high income. And here we have some the proportions of countries that are meeting all the minimum requirements by each of the core components that I introduced before, having a program in place, which was core component 1, having guidelines in place, education scored very low, and this was consistent with previous data and previous publications, which identifies that infection control in the pre-graduation is not standard and is not well established, and also having specialized education is still lacking. Surveillance, although scored high, however, since it is the minimum requirement, it requested very minimum requirements in the sense that what has been planned, and it did not assess their programs, the actual implementation of the program. An active program existed in at least 55% of the countries that were involved. However, there was a lower protected and dedicated budget when it comes to having the dedicated budget for the specific program. Having guidelines in place scored higher. However, the indicator which assessed implementation of these guidelines scored lower, and actions taken at the national level, at ministerial level, to support implementation or assess further follow-up of adherence to guidelines scored lower. Education, I mentioned, it scored low, and so the surveillance. But as I said, having a national strategic plan for surveillance was what was assessed. And here we have the proportion of countries with reported established minimum requirements. By color, you see each group of indicators within each of the 6 core components. I specified by minimum requirement to have an idea of what was assessed. So, for example, having an appointed focal point on infection control, having undergone training was identified here, and all the core components are listed. For example, having a national system for monitoring and evaluation of IPC training existed. However, it scored the lowest from the other minimum requirements that were assessed. As I mentioned before, one of the objectives was to compare with previous assessment that was done, and this was the very first national survey indicators in 2017-18, and the one that was conducted last year in 2021, and the indicators are listed in both of the assessments, since they varied slightly. However, we have seen some significant difference between the 2 cohorts. Having a national IPC program remained quite stable along between the 2. Having IPC focal person trained and dedicated professional there has been a large increase, and these might be needs to be studied further if this has been related, but an impact might have seeing the increase during the COVID pandemic might have influenced that increase. Having a dedicated and protected budget has also improved, and there has been in-service IPC curriculum, education, training. There has been quite some increase from 58% to 85.5% there. And this could also, we have seen throughout the COVID, and healthcare facilities and hospitals have provided massive training on use of personal protective equipment throughout the COVID pandemic. So that could have also influenced the increase. And also in surveillance, there has been in establishing surveillance in the national programs has seen an increase. So some of the yes, some of the points and challenges that come with such global survey, although it provides us with data which is and findings which are very challenging to obtain, it carries the risk of selection bias, since some active countries and national focal points that are more active are likely to respond, and that is also the self-assessment method, which is linked to the social desirability and the representativeness achieved from this global work. So just to conclude, there needs to be further implementation to strive and meet this full minimum requirements that have been established from these guidelines, to have active and functioning programs, not only on paper, but to have work plans and strategies that are in place, having dedicated budget for teams to work and perform the core components that are requested. Training and education is key, and investing in the expertise to conduct the monitoring and auditing. Thank you very much for your attention. Thank you.
Thank you all. And now we can start with these questions and answers. Those who have questions, can you write them or raise your hand? I don't know how this works.
Yeah, thank you, Carmen. For all participants, simply to know, you can either write your questions on the chat or simply open up your microphones and speak, whatever you prefer.
If there is no questions, just can I ask one question to Anthony?
Yeah, of course. Yeah.
Yeah, I have a question. It was very interesting. Maybe it's a little bit biased because I come from Brittany, but I would like to know, maybe I missed this information, but in your PopGen project, is it only healthy individuals or also people with pre-existing disease? And I have another question. Have you seen some association with the occurrence of some disease in specific area of Brittany and genetic variants?
So it's in general, we selected the individuals without looking at phenotype data. So they are essentially individuals of the general population, but they are all members of the existing epidemiological study Constance, which maybe you know. So in the future, we will connect the genotypes with the phenotypes to be able to do lots of association testings within this group, but they were not selected based on any. So essentially, it's a cohort of healthy individuals that will provide a control panel and to give a catalog of different genetic variants.
Okay.
For the second question, it's a very good one. I think there are definitely lots of people in our lab working on things like that. It's not something I've personally worked on, but I think there are certain associations with some. I think there's a congenital hip displacement phenotype that people in the west of Brittany have a higher frequency, and this is connected to some genetic variants that were identified more specific to individuals from the west of Brittany. And I know that there's work going on a slightly higher frequency of gastric cancers, and maybe when you have too much iron in your blood, I can't remember what it's called, A phenotype where you have a difficulty to metabolize iron in your blood. I can't remember what it's called. So there are a few which are being studied at the moment where there seems to be a kind of a local genetic effects. And but yeah, if there's no one else, I really enjoyed your presentation as well, and I definitely you mentioned that there could be some genetic factors playing a role, and I'm wondering if in the future you're interested to have genotyping data for some of the individuals in your clinical trials, and maybe some individuals.
I hope in the future, to have some information regarding the genetic background of this patient. But for the moment, I have not this answer. But in the future, we plan to do some experiments.
Oh, well, it'd be really a pleasure. You know, we should get in touch. Maybe one day. Who knows? We could work on that together one day. Yeah. So it would be very nice.
Yeah. Thank you.
Thank you.
So I have a question for Soizic. Because you've been talking about how you can predict the onset of virus, but it's like once you can do the prediction, how can you avoid them? Is there a way to avoid them?
It's a very good question. I think it will be very difficult to avoid this toxicity because you have the same with chemotherapy and other treatment. And here you play with the immune system. So of course, if you release some breaks, the immune system is over stimulated. So it's logical to have some IRS, and it's well accepted that if you have IRS, you will have a better response to treatment and a better outcome. But so the goal is really to identify patients with pre-existing disposition. So for example, some patients have some pre-existing autoantibodies. So if we can screen autoantibodies before treatment initiation, we can maybe better follow some patients and start maybe immunosuppression as soon as we can see some effect and not wait the final diagnosis of IRS. So the idea is to really to identify predictive marker to better follow patients with high risk of toxicities.
Okay, so you're thinking of a more personalized medicine with this.
Yeah, exactly.
Okay, thank you. So anyone who wants to ask any other questions? I have a question for Marcin if there are no more questions. My question for Marcin is if he thinks of a future in which we can have this common law that it's compatible with the individual laws of the different countries. What do you think?
So first of all, I wanted to indicate that in all those areas where these supranational laws issued and by EU exist, in many of these areas we are very effective. And the problem of infectious diseases is a cross-border problem in its nature. It's hard to think about many other issues that are so much disrespectable of borders than infectious diseases. And I think that especially in this aspect, our common European law should be more, I would say, vivid. I don't know whether it's good, but there is a treaty basis, especially in the Treaty on the Functioning of the European Union, to issue effective hard law, so-called hard law, not just the soft law recommendations or just guidelines to member states, but existing laws which will help to prevent spread of pandemics, epidemics in Europe. And as I mentioned during this presentation, there are such issues like European vaccination certificate. We had a big problem with this in Poland because, you know, of current politics and how the government looked for the polls. They decided whether it's good to have EU vaccination certificate or not. And certainly it didn't give a good results because we had the highest mortality rate in a certain periods in Europe, just because there were no common rules in this regard, I think. Because if there is a hard European law on this issue, then we had to respect it. Yes, so, and I believe that as a European Union, we have a much great intellectual, scientific, legislative potential to prepare good laws in these issues, yes? Because Poland has its own way of looking at this, Spain the other way, and Sweden the third one. We all have some good practices, but why not to take all of these together and prepare good piece of legislation and be the sample for other countries and regions how to do the good legislation in this regard? We can't hear you, Carmen.
Thank you. Sorry that I turned on my microphone. Now I have a question for Brendon. You were talking about these epigenetic mechanisms that affect how the immune system is trained. And I was thinking how, for example, the availability of methyl groups and acetyl groups affects this training, and because, for example, this would mean that macronutrients may affect the immune system in this way. So what do you think, Brendon, about this?
That's a great question, actually. And due to the constraints with regards to time here, I did not include immunometabolic aspects of both tolerance and trained immunity. From a lot of previous work also that I've done, we have seen that in the context of sepsis, for example, broad defects in energy metabolism characterize immune paralysis in septic patients, and this can also be reversed using cytokine therapy. But the underlying mechanisms tie into the availability of certain metabolites. For example, alpha-ketoglutarate is quite important for DNA methylation and all kinds of methylation itself. Likewise, itaconate. So I don't see a reason to discard micronutrients or any kind of nutrients with respect to engaging in these epigenetic mechanisms. And yeah, we do know that the food we ingest does have an effect on the way our cells or immune cells possibly, and I believe most likely also stromal cells and parenchymal cells respond. Yeah, good question.
Thank you, Ben. So if there are no more questions, I would ask my last question to Emilia. Like my question to Emilia is it feasible to implement these minimum requirements you were talking about in these low-income countries? I mean, how possible is it?
Very good question. And It is challenging, of course. However, the minimum, that is one of the main reasons why the minimum requirements have been then developed from this full list of core components, because they do take in consideration low-income countries. They have taken, and the consultation and the expert group that has been developed to create this minimum requirement has taken on board experts or focal points from low-income countries as well. So as I mentioned and gave a couple of examples, it did not, they are at the very minimum. They do not assess, for example, the surveillance, how far it has been implemented, but at least to have the strategy and to have, so I would say this point has been considered, and it is an evaluation after all as well that aims for countries and including low-income countries to further improve. So based on what they currently have and what can we do to support, what can the WHO do to further improve based on the gaps that are identified. For example, if they do not have specialized or trained experts in the field, if training is one of the problems, that area can be targeted by providing expertise for the resources and so on. So it aims to include, to be inclusive of all. Thank you.
Thank you. Well, I guess it's just we get to the end of this session.
I can ask a quick question.
So sure, of course.
A question for Brendon, in fact. So I'm really not at all expert in epigenetics, but it was a very interesting talk. I noticed you were looking at toll-like genes, and so something that we've looked a bit at is the stratification of these genes in European populations, and I think it's a gene where or a group of genes where there is some evidence that human populations have maybe benefited from incorporating Neanderthal genes into their genome. So I just, not really a question, just wondering if that's at all pertinent for yours. So maybe that these genes have helped us combat certain types of infections. Or just wondering if that's something you're looking at all, or if that's at all pertinent.
You mean genes themselves or variants within the genes?
Well, variants within the genes, yeah, of course.
Well, variants, genetic variants or germline variants, we haven't looked at those at all. However, from past, recent actually, studies in COVID patients, these are two back-to-back really important Nature papers, showed how, there is a potential genetic predisposition to severe COVID via what we know predominantly as interferon responses. I'm not sure how I'm not familiar with the chromosome, the Neanderthal section. I think it was some chromosome 3 or something. I'm not familiar with the specific region and the context of epigenetic shifts or any of the sort. Sensationalistic, right? The Neanderthal portion. But still, it's interesting, no doubt.
So any more questions? Well then, if there are no more questions, we can finish the session. And I would like to thank you all for the greatest talks that you've given. I think we have learned very much from your talks. We've understand that the immune therapy has its adverse effect, but it can be predicted. We've learned that the immune system can be trained, and we've seen how the different individuals in different population can be classified because of their genetic backgrounds. That then with seeing how we need to have these common laws to protect the individuals from infectious diseases, and we've seen also about infectious disease how there's these minimum requirements to prevent them. So I think it's been great decision. We've learned very much, and I would like to thank you all for your great presentations.
Thank you.
So, I don't know, Juan Ramon, if there's
Absolutely, Carmen. Thank you very much. I just wanted to say it again. Thank you very much to all of you, to all meeting attendees, to everyone who has been here with us today, learning and sharing knowledge, to our speakers, of course, because of your very interesting presentations and how you have found certain links or leads between your respective studies, and of course to you, Carmen, for your help organizing these and making this possible. For all of you, simply to know, this the recording of this event that has been live streamed on YouTube will be available there, and additionally each individual presentation will be uploaded, and a playlist will be created on YouTube so that your individual presentations can be directly accessible there. To all attendees, attendance certificates will be delivered in the next few weeks. We have been, and we are actually organizing several events in different initiatives within Research Use. So the delivery of these attendance certificates may delay a little bit, but you can be sure that they will arrive at some point. That's it. Once again, thank you very much to all of you, and see you at the next webinar of the CU Talent Initiative. Thank you very much.
Thank you.
Thank you. Nice to be organized.
Thank you. Bye.
Thank you.